All right. Hi, everybody. Sorry, I just had to wait for the coffee to finish percolating. Okay, where were we? This up there. Okay, let me just double check, make sure I got a picture taken of that. I think I did. What do you want? Oh, got a picture taken of my dog too. All right, so I'm gonna have to pick up Snowball. Come on then, come here. There you go. Oh, she can jump up there on her own. Easy peasy. Okay, so essentially these are all the different things that are going on. And remember, these are all going on kind of at the same time, right? So it's not that all DNA synthesis has to be completed before the RNA primers can be removed and the ligase can do its job. Pardon me. It's all happening at the same time. But these are all the different parts, players and processes, I guess, that are taking place during DNA replication. So you'll need to know all of these, right? And what they do and why. Ooh, nice cup of coffee. Okay, so eukaryotes. Obviously, eukaryotes are a little bit different. One of the biggest difference is that eukaryotes have linear chromosomes, right? So that kind of, well, and they're large as well. So we have to have multiple points of replication or origins of replication. It causes big issues with the ends of the chromosomes, and we'll get into why in a little bit. And also, eukaryote DNA is always complexed with proteins, right? So eukaryote DNA is actually nucleoprotein, right? So that's another word that's worth popping up. So that's DNA, oops, DNA plus protein, histones, actually. And that's also called chromatin, right? So that's stuff that you have to get rid of and then reassemble after replication. And you also have to copy those histones too, those proteins. You have to make more of them because you're making twice the amount of DNA. And so there's largely, by and large, things do the same things in eukaryotes as prokaryotes, but there are some little wrinkles. One of them is the DNA polymerases and the primases, or primase, right? Which are, there's a different way of doing it in eukaryotes. It's not a primase and then a DNA polymerase, it's actually a couple of different DNA polymerases. And also the removal of the primers, the RNA primers is different, but we'll get through all of those in a bit. So first up is, and we'll come back to this later when we talk about gene expression, because this is chromatin structure and the state of DNA and histone molecules has an absolutely massive effect on gene expression, right? Actually, it was funny. I get Science magazine, you know, the journal, as part of my, I think it's bibliotech. It's like San Antonio Public Library. You can have it on your iPad or your phone or whatever. And they mostly have like People magazine and stuff like that, which I'm not super interested in. But in science, they have Science Mag, which is really cool because it has really cutting edge, like high impact science in it. One of the articles that I read while I was eating my mini wheats this morning, which is actually true. I do eat mini wheats for breakfast. But anyway, while I was eating my mini wheats, was about what was it, neutrophils? It was about macrophage activation, which is really weird, right? So like how macrophages have different outcomes, you know, being immune response cells. And one of the ways is that macrophages sense the duration and intensity of the signal that they're receiving in order to have a different change in gene expression. And the way that they do that is by modifying this stuff, their chromatin, which is cool. Anyway, so that's a bit of an aside, but we'll come back to this in much more depth later on in the course when we look at gene expression. But for now, all you really need to know is that DNA is complex with protein, and each one of these is a nucleosome, and there's actually a little bit of fluffiness in terms of what actually is a nucleosome. This is what's called a core particle. And so the core particle is about 147 base pairs or whatever, you know, between 145 and 147 base pairs of DNA. So core particle, 247 base pairs of DNA wrapped around 8 histone proteins, which are actually 4 pairs of different histone proteins. So that's like the core smallest kind of unit essentially. And a nucleosome sometimes equals a core particle. This is where we get a little bit fluffy in terms of language. Sometimes it's from the beginning of one core particle to the end of another. Bless you. Which equals around 200 base pairs plus histone H1 protein. So language is a little bit fluffy about this and it can trip me up last semester when I was writing an exam because it leads to confusion. But basically the DNA is wrapped around a core of 8 histone proteins. How tightly that wrapping occurs depends on the state of those histones, which we'll get into later. That's part of epigenetics. And then that DNA is wrapped into varying degrees of compactness depending on state of the cell cycle and other factors, you know, where it is on the chromosome and likewise. Right. So one of the first things that has to be done in DNA replication is you have to hoof these things off, right? Because these will get in the way of DNA replication. And one of the in eukaryote DNA replication, one of the roles of helicase actually is to kind of essentially, like as it plows down the strand, is to kick out chromatin, which obviously isn't an issue in bacteria because they don't have chromatin, they don't have DNA and protein together. So that's one issue. And again, we'll get into chromatin in much more detail later on in the course. Second is that obviously things are more complex with eukaryotes, as always. There's a lot more DNA polymerases. And one of the other key points is that, so in bacteria, you have primase, which is an RNA polymerase, and then DNA pol 3 is the one that does most of the DNA replication. So, oops, in Prox, you have primase, which is an RNA polymerase, and DNA pol 3. In eucs, or eukaryotes, you have one DNA pol that starts which is alpha. And it has primase activity, but only, I think it does about 200 base pairs of DNA. And then essentially it falls off. It's like, pff, screw this for a game of soldiers. I'm off down the pub or whatever DNA polymerase alpha would say. Then it depends on whether you have it's the leading strand or the lagging strand. I don't actually even know how this is figured out, right? But when that DNA polymerase alpha falls off, if it's the lagging strand, delta comes in and does the rest of the fragment. If it's the leading strand, epsilon, which is that funky E, does the work. And so I don't know why. Presumably DNA polymerase epsilon is able to go the distance, right? It's like much longer processivity, maybe. But either way, the DNA polymerase that synthesizes DNA in eukaryotes depends on whether it's synthesized in the leading strand or the lagging strand. And also the removal of the primers is very different, right? We'll get to that in a sec. Anybody have any questions so far on any of this stuff? Cool. So in eukaryotes, instead of the DNA polymerase 3 falling off when it hits an RNA primer and DNA polymerase 1 coming in, removing the primer and then adding the new bases. Now what happens is we have our DNA polymerase delta, right, because it's the lagging strand, and it will hit the end of that primer, like the 5' end of the primer, basically. What happens now is we have this replication protein A, right, which is basically it binds single-stranded DNA, right? So this prime is kind of kicked out. These hydrogen bonds are broken. Replication protein A also brings in endonucleases. Essentially what it does is it cuts off that primer. So it doesn't degrade it base by base like DNA polymerase 1 does in prokaryotes. What it does is it kicks out the primer and then it cuts off a chunk, including a bit of DNA too, right? Not a lot, but a chunk of it. It's kind of actually more equivalent in a sense to mismatch repair, if you think about it that way. And then as soon as that obstacle is removed, DNA polymerase delta starts back up again and continues the synthesis of that strand until it hits the 5' end of the next fragment, the next Okazaki fragment. So again, a very different mechanism here. It's not so much a handover of activity as in prokaryotes, where DNA pol 3 stops, DNA pol 1 comes in and does its job. Now it's more like you kind of hit an obstruction, you wait for that obstruction to be removed, and then you continue on and finish the job. Now, the end replication problem is a pretty big one. And so in many ways, this is why we get old, which is always a sad thing to talk about because, you know, it happens to everybody, unless you die young, of course, which, you know, isn't much of an alternative. So I'm just going to stop sharing for a second because I want this bigger. So the problem is the RNA primer. So essentially what happens when you have your two strands of DNA, and we're just going to make this short. So we have 5 prime, 3 prime. Right, so this will be your, I don't know, original or template. And then when you go through your round of replication, obviously being semi-conservative, you have your, what color do I want to use? See, this is where you get into the problems with having 3 colors. You have your primer here, and your primer here. And then like so. Or, you know, if we're, this is, let's say, the ligand strand, we might have another primer here. All right, so far, so straightforward. However, what do you need for DNA synthesis? What's the key requirement other than having template and nucleotides? What else do you need? What do those nucleotides get added to? Yeah, they get added to the primase. Well, the primase makes an RNA primer and they get added to that. So what happens when you take away these primers? So here we have so you're going to continue filling in this bit like so. Where's the 3' OH here? There isn't one. Alexa, stop. Oh, geez. Alexa's just started up. Whoa, give me a second. So, essentially what that then results in is when each of these is used for the next round of DNA replication, you're going to have 2 which are like so. And then you're going to have the other copies, which are going to be using these new strands, which are like so. And these are supposed to be the same length. It's just a little tricky to do. And so over time, stuff toy out of there, your chromosomes will shorten by the length of the primer every round of cell division. So essentially at the, let's take a picture of this. And if we kind of zoom out a whole bunch, we have for each chromosome, and they don't look like ear, you know, cotton sticks or anything. We have our telomeres, right? And these equal repeat non-coding sequences. So essentially at the end of all of your chromosomes, you have buffers, right? Their job is to kind of absorb the chromosome shortening that happens via the end replication problem. So what happens when you get to the limit of the telomere, right? You kind of chew it all up. Does anybody have any ideas? What do you think happens when you've used up your telomeres? So I don't know how old all of you are, but most likely I'm older than most of you. My chromosomes are going to be shorter than your chromosomes because they've gone through more rounds of replication, particularly cells that undergo more cell division. And your chromosomes are going to be shorter than my daughters. My daughters shorter than, you know, their nieces and nephews, their cousins and stuff. I'm not very good at genealogy. Cousins, yeah, that's the right one. So what happens when that buffer is used up? What do you think? So this is a cool word. You get what is called, yeah, you can't copy your DNA anymore. If you can't copy your DNA, you can't undergo cell division. If you can't undergo cell division, your cell or that cell undergoes what's called senescence. Basically, that's cellular old age. So that cell can no longer divide again. Hello, trouble. And actually, it quite often starts accumulating kind of like what we'd call old age proteins, right? Have problems with protein aggregation and cell repair machinery and stuff stops working quite so well. It's not very well understood, but if you take this overall and look at an organism level, this is why you get old. Okay. Blame genetics. And so, and this is a really fun topic to talk about. I love talking about this. So each cell, so here's the rub, Adam. Each cell has kind of an intrinsic replication limit. That's called the Hayflick limit, which is around 60 cell divisions. So what about stem cells, gametes, germ cells, things like that? You're constantly making red blood cells, you're constantly making, skin cells, things like that. So how does stem cells evade that limit, right? And this is actually something that's really interesting because this is the double-edged sword of longevity, approaches to increasing longevity, right? So everybody wants to live forever, well, at least most people do, but no one wants to live forever and be really old and decrepit, right? That would suck because, being like age 80 for several hundred years, we're like, oh, my back. No one would enjoy that, right? You'd just be like, have a world full of like complaining old people that drive really slowly. So anyway, oh, that's a bit of a mischaracterization of old people, maybe. Oh yeah, yeah, no. I always tell my wife I'm an old man in training, and she just kind of rolls her eyes and goes and does something else. So for stem cells and germ cells, they can repair their telomeres, right? And really, germ cells, or the ones that generate the cells that generate germ cells, are in a sense stem cells anyway. They just generate gametes instead. And they do so using an enzyme called, unsurprisingly, telomerase or telomerase, right? And so what that does is it uses those repeats in the telomere and a bit of funky trickery to synthesize, that missing gap of DNA. And so they can extend their telomeres again. And so essentially, germ cells and stem cells are immortal, or the cells that generate germ cells, right? Stem cells can divide over and over and over and over again endlessly without ever hitting that Hayflick limit, right? They are essentially immortal. What other type of cells, and here is the double-edged sword, what other type of cells could you consider as being immortal, at least until the patient dies? What other cells can divide over and over and over again without limit? Cancer cells, Izzy, exactly. Good job. So tumor cells, well, yeah, skin cells is good. We want them to do that, right? But cancer cells essentially activate telomerase, right? And so they're able to escape that limit basically by activating the same mechanism that stem cells use to remain immortal, right? That's also why you've all heard of HeLa cells from Henrietta Lacks. Those were the first immortal cell line extracted. Now I've got to use punctuation there. From Henrietta Lacks. And obviously there's a huge kind of bioethics issue with that because she didn't actually give consent for her cells to be used that way. But up until that point, anyone that wanted to study human cells suffered from the issue that, you could kind of grow them in culture for a certain amount of time and then they'd die, right? They would senesce because any cells that you isolated from, non-stem cell tissue had an inherent built-in limit, replication limit, because of their telomeres. And so it made biomedical research very, very difficult. I think this was back in the mid 60s, I think, thereabouts, early 70s maybe. So when they discovered this immortal cell line, which came from, I think it was ovarian cancer that Henrietta Lacks had, those cells were able to replicate endlessly. And those cells essentially have led to a lion's share, like a large majority of the biomedical discoveries made using tissue culture. So they're the basis, not all of them, obviously we've got a lot more by now, but a large number of the cell lines that scientists use to study these things originally came from that one woman. And so in those cells, telomerase has been activated kind of inappropriately, right? Because we don't want cells to keep dividing over and over and over again unless they're stem cells. And that allows those cells to evade that Hayflick limit. So even though the story around Henrietta Lacks and her family as well and her descendants is pretty complicated, that's essentially what started kind of the biomedical revolution as well. Things like being able to make, insulin for diabetics and all kinds of discoveries that we wouldn't have made otherwise. So, and that all centers around, hang on a sec. Evie, your coffee's in the coffee maker. Can you clean out the filter and turn it off when you're done? Thanks. All centers around the end replication problem. The issue of those primers on linear chromosomes and not having something with which to fill in the gap once they're removed, right? Because you can't keep them there because they're RNA, you have to remove them. Anybody got any questions? Yeah. Anyway, now you know what the purpose of telomeres is. Essentially, it's a junk buffer, right? That you don't care about losing. Because you don't want any genes at the end of your chromosomes to start getting degraded, because otherwise weird and probably not wonderful stuff will happen as a result. Okay, so changing gears a little bit. I want to talk a bit about homologous recombination, and this is pretty much the end of the material for exam 2. So we'll finish this today. I think we've got enough time to cover it today, and we'll have a class period or thereabouts on Tuesday where we can go through any questions you have about the material, because the exam 2 will open on Tuesday at 1:30 or thereabouts. And so it'll be on all the material from beginning of this second section. Oh, couldn't hurt, right? To the end of what we're going to talk about today in terms of material. Obviously, we can talk about that on Tuesday for the first class period, but that's all that was going to be covered. So again, you're going to be doing a bunch of linkage stuff, guaranteed. You're going to have a lot of stuff on all of the different kinds of questions. You're also going to have a lot of kind of molecular type, just recall, what does this do? What step does this play a role in? Things like that. So it's going to be a bit of a mix. And it'll be a transition too, because the next 2 kind of quarters of the course will be much more molecular biology focused. All right, so this should give you a preview of what it will be like for exams 3 and 4. Actually, before I forget, let me take a picture. I just think the word senescence is such a cool word. Not many times you can actually use it in everyday conversation, but nonetheless. Okay, so Homologous recombination is generally something that we're concerned about with regards to meiosis, right? So particularly, well, it occurs in prophase 1. So this is what generates the recombinants, right? The new combinations of alleles in the gametes that didn't exist in the parents of that individual. Right? It's also a high fidelity way of fixing DNA damage. And so this is something that can happen during mitosis if DNA damage occurs to one of the sister chromatids. Oh, actually, give me a second. See if I can find it. Good picture. Not sure if I'm spelling this right. This is often called Conan the bacterium, which is kind of fun. So this is a bacterium. It's a very funky bacterium, actually. Looks a bit like that. Actually, looks just like that. Ta-da! So it kind of exists as like a 4-cell clump. Actually, looks a bit like sure what they call those, like Italian dinner rolls. You know, when you get them in a packet, looks a bit like that. Makes me think of lunch, actually. Anyway, so these are able to withstand about 500 to 1,000 times the radiation, gamma radiation that will kill us, kill a human. So they are astonishingly radiation resistant bacteria. Like they can actually live, I'm not sure if these ones particularly live on uranium, but others that are similar can actually live on, you know, uranium as a substrate, which is kind of crazy if you think about that. And so it does so because it has multiple copies of its genome at any one time. It's not more resistant to radiation. It just has a much better way of fixing those errors than we do. And it can do that at all times as well. So yeah, this is called Conan the Bacterium. Has anyone actually ever seen Conan the Barbarian? It's probably a little bit too old for you lot. Arnold Schwarzenegger. Let's see. Look at that. What a beefcake. Don't know if any of you are particularly fond of well-muscled guys covered in oil. Not really my thing, but if it were, here you go. Anyway, that's a super cheesy film from like the 80s or something. Anyway, so a little bit of a tangent, but just to illustrate that this is also a very good mechanism for fixing double-stranded breaks, which are usually pretty catastrophic. So this can happen during, where's the chat box gone? Get in the chopper. Where does that come from? Don't think there are any helicopters in Conan the Barbarian. Oh, yeah, right. Yeah, good stuff. I got to watch that with my kids sometime. They kind of completely missed the whole Terminator series. So homologous recombination can occur in meiosis, or occurs, right? Not can. That's prophase 1. And kind of between S and G2 phases in mitosis. So the key requirement for homologous recombination is you have to have either a homologous piece of DNA, which is the case in meiosis, right, the homologous chromosome, or a sister chromatid, and this is in mitosis, with which to use as a template, right? So this is a key critical requirement. You have to have some kind of DNA there that is close enough match to the bit that needs fixing that you can use it to kind of line up the broken ends and fix it, right? So in meiosis, homologous recombination is induced, right? It's done deliberately. In the case of DNA repair, it's in response to a double-stranded break. So for example, from gamma radiation, which breaks the DNA backbone. And so, dang it, my chat box has disappeared again. Super annoying. There we go. And so one of the key things is that you have to line up your DNA perfectly, right? Absolutely and totally perfectly. Because if you don't, you'll end up basically with one finger longer than the other one, right? You won't have a precise exchange or copy or fix, depending on the process that or the purpose of that homologous recombination. So you have to line things up perfectly. And then you actually, if a double-stranded break doesn't already exist, you have to induce one, right? So you have to break the DNA and then you do a whole bunch of funky stuff. And at the end, what you want is two molecules, which are most likely going to be recombinant, but don't have to be, which have not lost or gained any information. Right, so they're kind of perfect repairs, essentially. And so just as an aside, right, so HomRecom is one form of DNA repair of DSBs, which stands for double-stranded breaks. So that only is going to happen if you have matching DNA to use to fix that break. Because if you imagine if you've got a piece of DNA and you break it, well, if you only have two free ends, it's pretty easy which end gets stuck to which, right? What about if you have two breaks, two free ends? Now you could just end up sticking the wrong end to the wrong bit, right? And that's what happens if you don't have a template available. What you get is, if you don't have a template, you get what's called non-homologous end joining. And really what that in shorthand or vernacular is a Hail Mary, well, let's just stick this stuff together, right? Even if it's wrong, that's better than, the having to die because our DNA is broken as a cell. So it's very much a Hail Mary fix, and it is not a very accurate one. And so when you look at kind of evolutionary history and you see kind of chromosomes getting stuck together or chromosome rearrangements and stuff like that, quite often it's because of DNA damage and the fix not being very accurate. Also, in a lot of cancers, you actually see this too. It's called chromosome shattering, which is kind of astonishing. It's basically a chromosome just breaks into lots of pieces, and then those pieces just get stuck back together willy-nilly, which causes all kinds of issues. So anyway, we're not going to talk about fixing DNA damage anymore with regards to homologous recombination. We're just going to be talking about the process during meiosis, right? Prophase 1. But just kind of keep that in mind, in terms of DNA repair. It's kind of an interesting topic to think about. So there are a couple of different models for homologous recombination. I'm going to talk about one because it's kind of a little bit more straightforward to talk about. So essentially what happens is you have to line up the chromosomes first, base by base. And this is what the synaptonemal complex does. And it has to be perfect because otherwise you'll gain or lose information in the recombinants. Now, once you've done that, then one of the strands has a double-stranded break induced, right? So you have to break the DNA for recombination to occur. And obviously that's not something you do lightly. And so the idea here, and like the kind of general big picture kind of idea, is we're going to cut that DNA back. So we're going to end up with single-stranded DNA for each end of this broken chromatid, right? So we're going to chew back some of the DNA so we have essentially overhangs, right? Single-stranded bits of DNA for both, probably better like that, both strands. And then we're going to use the other chromosome, chromatid, as a template to continue the synthesis across this gap, right? That's the general kind of idea that we're dealing with here. So again, break it, chew it back, use the other double-stranded DNA molecule as a template. And this is where it gets a little hairy. So we've broken the DNA. Now we're going to chew back using an endonuclease, right? Which just not an endonuclease, an exonuclease, sorry. The endonuclease is what cuts the DNA in the first place. Exonuclease is going to remove from the 5' end of each strand some of the DNA. So you end up with single-stranded DNA for a stretch either side of that break. Once you have that single-stranded DNA, we get what's called a D loop, right? So this strand kind of gets yanked out. It gets pulled out. And the reason is this strand here, let's say the one on the right at the top of the blue strand, it invades the homologous double-stranded DNA molecule. So because these are, if they're not identical, they're nearly identical, right? They're homologous chromosomes. This 3' end, this free end is going to match the other strand of this double-stranded intact molecule, right? They're going to base pair because they have the same DNA sequence. And so now you have a bit of single-stranded DNA here, right? Just after this strand is invaded, and you have a 3' OH. So now you can begin DNA synthesis. And this is the kind of the follow-on part of homologous recombination. It always involves new DNA synthesis. And I make sure I'm keeping everybody with me. I don't want to leave anyone behind. How's that doing for making sense? Yeah, it's tricky, particularly this this particular part of homologous recombination. And I'm not going to lie, it took me a couple of times of teaching this to kind of fully twig and get comfortable with it enough to teach it to you lot. So in a sense, that's how it's a little easier for me to teach it, because I know what's difficult to understand, because I went through that myself, you know, within the last couple of years or so. So again, this strand up here, this free single-stranded bit, right? So we don't have two strands here. It's been chewed back, so we have one kind of free end, kind of like flapping around in the breeze. That weasels its way in to the homologous strand, homologous DNA molecule, and base pairs with its counterpart in the other chromosome, like so. Now that it has done so, right, so we've got, you know, base pairing along here, hydrogen bonds, so on and so forth. Now it can be used essentially like DNA replication, right? It's going to use this homologous strand as a template, and that invading strand is going to get extended as if it was, you know, synthesizing a new strand of DNA. And so that kind of continues along, right? It's a very pushy, invasive strand. So it continues for a stretch, and essentially it needs to continue to the point where it can get matched up with the 5' end of the other broken fragment. Okay, so that does one strand. What about the other one? So the other one is using this D loop, right? This stretch of bit of DNA that has been forced out, right? So it's single-stranded, flapping around in the breeze, right? Doesn't have any DNA paired up with it, but it is exactly complementary to this free end of DNA from the broken part. So you can see that now, okay, we've still got this invading strand over here, right? It's being extended, forcing out the other strand of the homologous chromosome, this red loop that's looping down here. However, after a certain point, that loop is going to be close enough to pair up with the other free end of the broken strand. And then we have the same set of kind of conditions, essentially, right? We have a template strand, this D loop that's hanging down. We have a free 3' OH. Now DNA polymerase can assemble and synthesize or can continue that strand from that 3' end of the broken bit down here. Right, so now you can see, and essentially the orange is showing you the newly synthesized DNA, which really, if you think about it, is going to be matching up to the bits of DNA that were originally here, either side of the break, that got chewed back by the exonuclease. Right, so you break it, you chew it back, and then you kind of continue, you know, you remake it using the intact strand as a template. And so once that's gone on far enough, essentially each strand will hit the other broken end, right? So this lower strand will continue to be synthesized until it hits the 5' end that was chewed back of the other half of the broken bit. And the same up here will synthesize this until it hits the 5' end of its counterpart on the other side of the break. Once that happens, DNA ligase comes in, sticks them together, and now we have this kind of funky hybrid, right, where we have kind of one kind of intact normal DNA strand, essentially, and we have two bits which are shared, right? So we have one bit of DNA which consists of, you know, half and half of original and the newly kind of synthesized or fixed, and vice versa on the other side. And so where these strands cross over, these are called holiday junctions. And when you actually look at it in an electron micrograph, that's what they look like here, right? So this is essentially the equivalent of what you can see on this graphic. It looks like a bubble in the DNA. And typically you have 4 kind of DNA strands kind of radiating out from that bubble. That looks like this. So this in here is going to be an original, and it's kind of hard to figure out just from looking at the picture. There'll be an original strand on both sides, and then one of these strands will have a bit of the other one, essentially. And these are what help hold together the homologous chromosomes during meiosis, right? These pairing with the other chromosome. And so these holiday junctions have to be cut, right? They have to be fixed, because otherwise you're never going to be pulling apart these homologous chromosomes in, what would that be? That would be in meiosis 1. So these are resolved by an unimaginably called holiday junction resolving enzyme. And so they will cut the DNA and basically stick it back together so that those chromosomes can separate. And depending on exactly which bit of the DNA they cut, whether it's crosswise or vertical, or even kind of left and bottom and top and right, or whichever, is going to generate different combinations of DNA. So it's going to generate recombinants, or rarely it generates non-recombinants as well. But you'll end up with 2 chromosomes which consist of a bit of the other chromosome. And that's homologous recombination. There's obviously a lot more to it than that. And there's also the second model, which is called, I think, single-stranded strand assembly or something like that. Single-stranded strand assembly. I can't remember what, SSSA. But I don't understand that well enough to try and teach it to you, and I don't want to confuse things even further. So, anyone have any questions? So I'd suggest going through all of this. Again, practice that linkage stuff so that it's like that. Your 2-point and your 3-point crosses. Don't forget all the other material. Yeah, mine too, Adam. Right, so we have all of the non-Mendelian inheritance, linkage, DNA replication, homologous recombination are what will be on exam 2. So there's obviously a bit of a spread there between kind of more kind of inheritance genetics and more molecular biology. And the next section, section 3 and 4, will be much more molecular biology based. We're going to move away from the inheritance genetics, more into the, or transmission genetics, I guess, more into the molecular genetics. Cool. So I guess the other question is, do you want to meet at 2 and have a long lunch break, or do you want to meet at 10 to 2 and finish earlier? What was that again?
I'd rather finish early, you know, but
right.
I don't know what everyone else wants to do.
Cool. We got 2 votes finishing early.
Okay, good.
All right, that's 2 out of 2. Representative democracy. Cool. Yeah. Okay, that sounds good to me. So let's meet at 1:50. And enjoy your lunch, and we can talk about the mini lab report. Nope, you were outvoted. No, we're going to meet a little bit earlier so we can finish earlier. So because we finished lecture 10 minutes earlier, we'll start lab 10 minutes earlier. Cool. All right, everybody. I'll see you in a little bit.